When researchers test a new medication, they run it through a randomized controlled trial. The explicit goal of this process is to measure the drug's exact biological impact by mathematically subtracting out the placebo effect. Over decades of clinical research, this subtraction method created a professional bias. Modern medicine began viewing the placebo effect as an adversary, statistical noise that had to be eliminated. This chart shows the total treatment effect is an additive combination of factors. The top blue section is the specific drug effect, responsible for only a fraction of the outcome. The three red blocks beneath represent the placebo effect, driven by psychosocially triggered physiological changes. This placebo effect serves as an active, integral component of any medical treatment, functioning as a predictable and measurable biological variable. This forces us to ask a structural question. Why do identical pharmacological interventions given to similar patient populations frequently yield strikingly different clinical outcomes? The variance occurs because the placebo response physically recruits disease-specific biological networks, such as the immune system and the endogenous opioid system. An inert sugar pill cannot induce these changes on its own. The physiological shifts are triggered entirely by the patient's internal psychological processes. Those internal psychological processes are directly constructed by external inputs, specifically the physical environment of the clinic and the behavior of the medical practitioner. This flowchart details the exact causal chain we are going to map. We will trace how a brief social interaction with a physician informs psychological processes, which then translate into a quantifiable neurobiological healing response. The sequence begins before a medication is ever prescribed. The process is initiated by the social context of the clinical environment itself. Psychologists use the term thin slicing to describe our cognitive ability to form highly accurate inferences about a situation based on incredibly narrow windows of observation. Within the first few seconds of a clinical encounter, patients automatically thin slice their environment, making immediate evaluations of their medical provider. During this rapid assessment, the brain isolates two specific traits: warmth, which measures the benevolence of the provider's intentions, and competence, which gauges their medical ability. This calculation relies heavily on nonverbal cues. The physical proximity of the physician, their body posture, and visual symbols of authority feed into this calculation. This split-second social evaluation serves as the primary input variable. It dictates whether the downstream neurobiological placebo response will be activated or dampened. Once the social context is evaluated, that data feeds directly into the patient's internal psychological processing center. A major component of this processor is implicit learning, the non-conscious acquisition of knowledge. This includes classical conditioning to the routines of medical care. Over years of doctor visits, objects like blood pressure cuffs, stethoscopes, or even the specific color of a pill become implicitly associated with recovery. These associations build specific expectations, like the belief that a certain pill will relieve pain. But those individual expectations are governed by a much broader cognitive framework. This larger framework is known as a mindset. Mindsets are broad cognitive scaffoldings that orient an individual. For example, a patient might hold the underlying belief that their body is fully capable of managing an illness. When a provider successfully signals warmth and competence, they directly shape these broad mindsets, elevating them into highly functional psychological frameworks. These mindsets act as the primary catalyst for objective physiological work, manifesting in measurable biological changes. This image displays localized metabolic activity in the brain. When placebo responses are triggered, we observe specific, quantifiable changes in these targeted neural regions. The process recruits non-opioid neurotransmitters, triggering dopamine release in regions governing motor activation and reward. Simultaneously, it activates the endogenous opioid system, which directly regulates the body's internal pain modulatory network. The effects extend well beyond the brain. Placebo responses trigger peripheral alterations, including measurable modulation of the immune system and suppression of lymphocyte cell counts. The psychologically constructed mindset serves as the chemical switch, turning on these precise, measurable neurobiological mechanisms. We can observe this entire sequence operating in controlled clinical data, specifically in a histamine study conducted by Howe and colleagues. Researchers induced an allergic reaction in participants using a histamine skin prick. They then treated the reaction with a completely inert placebo cream. The independent variable wasn't the cream, it was the practitioner applying it. Researchers deliberately manipulated the provider's display of warmth and competence across different patient groups. When the cream was administered by a warm and competent provider, patient expectations successfully activated the biological mechanism, resulting in a physically reduced allergic response. However, when the exact same inert cream was applied by a cold and incompetent provider, the physiological placebo effect was completely negated, yielding a severe allergic reaction. The data confirms the model. Identical inert treatments generate distinctly different physiological reactions based purely on the social context of the person applying them. This flowchart reveals the completed causal chain. Thin slicing a social environment informs a psychological mindset, which in turn activates a biological mechanism. By mapping this horizontal causal sequence back onto our original view of clinical treatments, the relationship becomes clear. The exact structures of the placebo sequence form the foundational physiological blocks that operate underneath any active drug effect. This means the clinical utility of the placebo effect does not require a physician to deceptively administer saline drips or sugar pills. Because patients are continuously thin slicing their environment, a practitioner can transparently and intentionally shape a patient's mindset regarding their disease. Optimizing bedside manner calibrates a quantifiable neurobiological variable, maximizing the total clinical efficacy of the active drug. Medical professionals do not have to prescribe a placebo to harness the placebo effect, because ultimately, optimizing the variables of the human clinical encounter is sufficient to activate the biology of healing.